Okay. Now, for Scispace, the name is Scispace. Unlike consensus, where you just type consensus.ai and you start. For Scispace used to be known as typeset. So what you do is to type typeset.io for you to be able to get to Scispace. Is that clear? I've given.
Say it again. Say it again.
Check the meeting chat.
Yeah.
I've given the link for you to go to Scispace there by clicking on that.
Typeset.oi.
IO.
IO, yeah.
Yes, yes. So by clicking on that, you get here. Now, the things we did with consensus, we can do it here, get insights about paper. The other aspect I want to teach us here, which is more, just like you can type your questions here, ask, and it will give you insight about paper, just like we did for consensus. But what I'm more interested here that I want to teach us is how to extract information in real time from a paper. You can, of course, when you get here, you have to sign on. I already have signed in, so I have an account. This one too used to be completely free, but now they have made an upgrade version that for you to be able to access certain things, you must pay for the upgrade version. But you can still achieve a lot with just the free version. You can do your literature review. You can extract data from PDF. You can read with Copilot. Copilot is an AI tool, and you can do your paraphrasing here too. You can do paraphrasing. You can do citation generator. Maybe you have a document, you don't have how to do the citation. You can click here, you can do citation generation for you. And let's say you enter the URL and click on search, it will give you the citation generation, how you should write the citation. But that's not what we are interested in here. And do paraphrasing for you, just like you have in Killbot. And the advantage of this, Killbot too has a paid version. We are all using the free version. The free version can only allow you to do paraphrasing in standard and fluent. It can allow you to do more. But for Scispace, you can do paraphrasing in several: academic, fluent, formal, creative, friendly, boldly, persuasive. All these are free. Assertive, supportive, empathetic, condescending, enthusiastic, so much. They are all free. But what I'm most interested in here is, for instance, if you have gone to consensus, you saw a paper on malaria vaccines, for instance, and you get the full text of the paper. Oh no, this one is paid. I'm looking for the one we can get the PDF version. It's open access, yeah. PDF. Yes. Now we can download this. Journal of Internal Medicine. Now, let's go back to Scispace. You can upload, I've downloaded it before. You come to this upload PDF. I go to download the journal of, and I click open. I upload this file, click on upload. This is the paper here now. If you click on this paper on Scispace, if you click on this paper on Scispace, of course, it brings it back here. And you can use Scispace to ask questions on this paper and see what it can do for you. For instance, this paper, I want to know what is the practical implication of this paper. What is the practical implication of this paper? Looking at this paper, what are we going to gain from it practically? And I click on this. Okay, it has not fully loaded this. Let me pick something I've used before, and something that's on my library before. Let me pick, okay. This is one of my publication on my library. In fact, let me tell you people here. I delivered my inaugural lecture not too long ago. And one of the things I did was that I fed Scispace with all my publications. And I started asking Scispace to summarize my publication for me, to tell me important findings in my publication, so that instead of reading through all my publication, then picking out what I want to use, it was Scispace that was giving me those information, and that was how I was able to quickly write my inaugural lecture. For instance, this, I want to find out, there's some, if I want to know, there are some questions that are already here. I can pick any one of them. If I say, what are the contribution of this paper, for instance? Scispace gives me the answer immediately. The paper investigates the antimicrobial susceptibility pattern, plasmid profile, and presence of resistant genes in Salmonella strains isolated from raw meat and stool sources in Niger State, Nigeria. The study aims to determine the distribution of resistance genes of ciprofloxacin and ampicillin, which are relatively high and low-cost antibiotics, respectively. The research provides insight into the prevalence of antibiotic resistance in Salmonella isolates. The study highlights the widespread distribution of antibiotic-resistant genes, especially to low-cost antibiotics. The author acknowledges the valuable contribution of staff, blah, 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 of Germany and so on. Now, I can ask further question. What is the antimicrobial susceptibility pattern? The study determined the antimicrobial susceptibility pattern of Salmonella isolate from raw meat. The results show that Fluoxacin and ciprofloxacin were the most effective antibiotic with 67.7% and 93.5% of the isolate being susceptible to death, respectively. Salmonella isolate exhibited multiple resistant patterns to ceftazidime, cefuroxime, ceftriaxone, erythromycin, ampicillin, clozacillin, and gentamicin. The study highlights the incidence of antibiotics. So I can ask question like that from papers, from any paper. So you can ask real-time question. Somebody should suggest a question I should ask based on this paper so that I ask it and I will see the result. Suggest a question based on this paper that I've loaded now that I can ask it. Suggest a question in the chat room. Are we together?
Yes, sir.
Yeah, somebody should please suggest a question, please. Okay. What is the gap in these studies? Everybody should mute, please. Everybody should mute. See, also, the study does not explicitly mention the specific gap in the research. However, based on the information provided, some potential gaps can be inferred. The study focuses on the prevalence of antibiotic resistance in Salmonella isolate from raw meat and stew sources in Niger State, Nigeria. It does not explore the factors contributing to the development and spread of antibiotic resistance in these sources. The study does not investigate the genetic mechanism underlying antibiotic resistance in Salmonella isolate. It only mentions the presence of resistant genes, Blatem and Gyre-A, and plasmid without further characterization. The study does not provide information on the source or origin of the Salmonella isolate, which could be important in understanding the transmission dynamics and potential sources of contamination. The study does not assess the impact of antibiotic resistance on human health or potential for transmission of resistant strains to humans. You see, very beautiful gaps that somebody can do further studies on. Somebody wrote, what is the contribution of this knowledge of this study? What is the contribution to knowledge? The study contributes to knowledge by providing insight into the antimicrobial susceptibility pattern of Salmonella isolate. It highlights the prevalence of antibiotic resistance. The study identified ofloxacin and ciprofloxacin as the most effective antibiotics against Salmonella isolate. The information is valuable for the understanding of local antimicrobial resistant pattern in Salmonella and can aid in the development of appropriate treatment strategies for infection caused by this bacteria. The study also acknowledges the valuable contribution of the staff of Molecular Biology and Biotechnology Department, Nigerian Institute of Medical Research, and Alexander Van Humboldt Foundation. That one is not important, but yeah, that's contribution to knowledge. What are the methodology used in this study? What is the methodology used in the study? The study involved the screening of 98 samples, including 72 raw meat and 26 stool samples, for presence of Salmonella species. The antimicrobial susceptibility of Salmonella isolates were determined using the Kirby-Bar disk diffusion method with 10 commonly used antimicrobial agents tested. Plasmid profile of the isolate were analyzed to determine the presence of plasmid, and PCR amplification was performed to detect the presence of beta-lactamase and gyre-A genes. The study also reported the molecular sizes of plasmid detected ranging from 6557 base pair to 23137 base pair. So, please, everybody should mute. This should not be difficult to do. Somebody wrote, what percentage of the tested population was positive for Salmonella species? Percentage of tested population positive for Salmonella. Among the 98 samples tested, Salmonella species were detected in 31 samples, resulting in a prevalence rate of 31.6%. The prevalence of Salmonella in meat sample was 30.6%, and in stool samples was 34.6%. The distribution across different locations in Niger State was as follows: Bida 41%, Lapai 41.2%, Mina 23.5%, Suleja 29.4%, Kotangora 30.8%, Wushishi 17.6%. So this gives beautiful rendition and insight to the paper. What are the limitations of these studies using simplex PCR assay for this study? If you're asking for using simplex PCR assay, you are asking for an opinion, because what we used in this study was simplex PCR assay. So if you are saying, what is the limitation using this? You are asking for an opinion, which is not based on the paper. So, and its answers are such: the provided sources do not explicitly mention the use of simplex PCR assay in the study, so it is not possible to provide specific limitation related to the technique. However, in general, some limitation of using simplex PCR assay techniques. So it now gives a general overview, but not from this paper, based on this paper. So we always have to streamline kind of question we ask. Yes, ask empirical question. What is the relevance of this study to the global community? The study is relevant to the global community as it addresses the global evolution of antibiotic resistance, which is a major concern worldwide. The investigation into the prevalence of antibiotic resistance and the role of plasmid in Salmonella strains is crucial in understanding the spread and mechanism of resistance, which can have global implications. The study provides insights into the antimicrobial susceptibility pattern of Salmonella isolate, which is important for guiding treatment options and controlling the spread of antibiotic resistance globally. Understanding the distribution of resistance genes and plasmid in different environments, such as raw meat and stool sources, can contribute to the knowledge of how antibiotic resistance spreads and can inform strategies for prevention and control on global scale. The findings of this study can contribute to the global efforts in monitoring and combating antibiotic resistance, especially in developing countries where the prevalence of resistance gene and low-cost antibiotics is widespread. Note, the answer is based on the information available in the provided sources, which is this paper. So this is the beauty of Scispace. So what you can do with consensus, you can do some of them here too. But this additional thing is what Scispace brings that you can add to what you do in consensus. For instance, what was the question we asked earlier? What is the role of proteins in vaccine development? We can add Scispace too. And just like consensus, here it gives insight from 5 top papers. So proteins play a crucial role in vaccine development. Initially, vaccines consisted of live or inactivated organisms, but it was later discovered that components of these organisms, such as proteins, could be used as vaccines. Adjuvants are often required to enhance the immune response. Recombinant DNA technology has allowed for the synthesis of well-defined proteins, like those of the surface of virus and bacteria, which can induce strong immune response. However, the development of protein-based vaccines has faced challenges, and many have failed to provide protective immune response. Despite this, recent success with recombinant protein-based vaccines for diseases like meningitis and shingles suggests their potential utility. Protein-based vaccines are also playing a significant role in the COVID-19 pandemic, with most COVID-19 vaccines based on the SARS-CoV-2 spike protein. Proteomics and structural proteomics have become important tools in vaccine development, aiding in identification and characterization of novel antigens and contributing to design and management of vaccines. So it now gives some papers an insight of each paper, just like we saw in consensus. It gives insight to each of the papers. So you can read from here. The role of structural proteomics in vaccine development, recent advances and future prospects. Although this paper might be relevant, say the paper does not directly mention the role of proteins in vaccine development. However, the review describes MS techniques, structural proteomics, and illustrates structural proteomics to vaccinology potential application. So it gives insights into what each paper says. And from here, you can sort by relevance. You can sort by citation count. So those that are highly cited, you pick first. You can also sort by newest first. The most recent will be the one to come out first. So this is a 2023 publication on the topic. And in this 2023 publication, protein act as the main constituent in peptide-based vaccine, where the main component causing the infection is deleted by recombinant DNA technology, and peptide acts as antigens to stimulate immune response. So this gives great insight into the paper, and it's a 2023 paper. So if you are looking for recent paper, then if you are looking for a very old one, so in essence, you want to write a review paper on a particular topic, you can sit here on Scispace and completely write your review. Completely write your review, because you can get as many people as possible from this place. And you can then pick out each paper, download them, upload the PDF into your library, and begin to ask various questions. And a co-pilot will be answering you in real time, just like we have done. So that's basically what I want you to know on Scispace. There are other things you can do from Scispace. Like I've told you, you can do your citation generation. You can do paraphrasing. If you have paid one, you can do AI detection too. So that basically is what I feel you should know on using your Scispace.
Thank you so much. We so appreciate this practical session. What do we say to Professor Dixon? Just type it on the
Thank
chat
you, sir.
box.
You, Prof.
Start your
God bless
response
you.
chat box. I'm happy that he had the time. See how much time he has spent teaching us practical things.
Thank you, Prof.
You can type your questions because we are ending now. Type your questions in our platform. Then I'm going to send us a Google form. 1, to evaluate what we have done in the 2 classes. 2, to determine what other class we are going to do next. We have other classes we are going to introduce to you, and then we will choose. Those that have the highest frequency, we will bring them forward and bring others backwards. I'm sure that this will help you. Then, before we go now, because I intended us to stop at 2 o'clock, you will have an assignment, which is a full review paper for publication. Your timeline is between now and ending of January. You post your full paper on our platform. I will set up a team within us to review your paper and assess it for publication. Once it is correct, we will publish it. We will tick it for you in the platform. All right? The essence of this is to make sure that this training is not a wasted training. We want to see you climb, we want to see you move, so that we can simply say to others, we have the best team. Or what do you say? Are you with
Yes,
me?
Prof. It's a good one. Yes, sir.
Yes, Prof.
So please, between now and end January, you must submit your assignment. Choose topic in your various areas. It's a complete review. All right. The three things you have learned now: systematic review, consensus, and Scispace is enough to give you a high standard review paper, which must be published. When you are ready, we'll start looking for good papers to publish, those that can publish for free, those that have small money to pay, then you publish. Want to tick it for you as your publication goes on. That's that for now. So if you have questions based on what we have studied now, the few minutes remaining, just type your question and then our resource person will answer you before he will leave finally. You can type your question now.
Okay. Prof, one thing I could also say is that if you give between now to the end of January, the time to do this thing is actually too long.
Okay.
With consensus and Scispace, if we dedicate 2 days,
1 week,
2 days, we can write a very good paper. And then we'll now keep tweaking. If we give 2 weeks, that paper should be ready.
Okay.
Yes. So that if you give too long a time, people might be relaxed and wait till the end of that when it's almost.
They would have forgotten the skills they learned.
So our
Okay, fine.
advice, they start immediately. And in the next 2 weeks, the paper should be ready.
Fine, I accept. So your time has been reduced to 2 weeks. 2 weeks is about 13th to 15th of January. So note it down. Any topic of your interest, depending on the area your research is going, choose your topic and go ahead and write. Submit in 2 weeks' time, between 13th to 15th of January. That will be okay. Then we will set up our next training for January. Is that okay?
There's a question which I want to quickly respond to.
Yeah.
It's talking about where to let me read the question again. Where can we find the library to add up any downloaded journal? If you go to home, you can see my screen. You can see where the library is. I'm clicking on the library now. Click on it. You see the upload PDF here. You click on upload PDF. It takes you to where you have stored your PDF, and you select, you click open. Then you come here, you click upload file. You see, this is the file I just uploaded. So that's how to do it. It's very simple. There is no trick to it. Very simple thing. I want to know about systematic review, which has not been taught here. Systematic review has not been taught.
Systematic review has been taught in the first class, unless you didn't attend, the one taken by Dr. Morris. And the slide is in our platform, unless you were not in that team. Yes, somebody has a hand up. I think that's Hannah.
Good afternoon, everybody. Can I be heard?
Yes,
Yes,
we can hear you.
But you have very short time to ask your question.
Okay, Prof, please, can we have like some notes on these things? Because like the systematic slides I looked at, those slides were not like notes. They were just points to be discussed. The actual discussion is not on those slides. So I didn't get anything from looking at the slides. And then today again, we have gone through another training and there are no notes. There's nothing to make reference to. We can, like me, let me speak for myself. I can open this Scispace in 30 minutes from now, and I'll be wondering what to do on it. It's not everybody that is that, smart to be able to know all these things just by being told what to do. So I don't know if we can have like a very brief note on both methods, the systematic and this one, and how to navigate this stuff. Thank you.
Sorry, let me just interject. The space is closing now. Let all of us log in back so I can handle these questions briefly. All right?
Yeah, let me answer one question here before we log out, please. Oh.
Go ahead.
Okay. Adenike said, I perceive it to be difficult doing a fast review without a paid version of consensus and Scispace. That's not true. You can do everything you want to do with the free version. So that's not an excuse. Adenike, can do everything you want to do with this free version. And is it Hannah that asked the last question? Yes. The issue is this. After we finish this, go back and start using it. In fact, you will learn more things than what I've told you. Just start using it. There's no trick to it. Just start using it and you'll begin to discover things. But most importantly, like I will always say, use AI as a research assistant, not as your supervisor. Don't allow AI to think for you. You know what you want, and ask your research assistant to do the dirty work for you. But you are the supervisor of the AI, so you must know what you want before you ask questions. So you must streamline all your questions to what you exactly want, not what. Don't just ask open-ended questions and expect to get good information from AI. Thank you, Prof.
Thank you. So are we coming back? I think all the questions are answered. So if you have more questions, type it in our platform, and then the Google form will follow later.